All right, now it's 12:48. Let's see, Fengchao is there. I think Kevin is also there. I don't know if Olga is going to be joining us for now, but I think we can start, right?
That sounds good to me.
All right, so let's see here. So I'm going to start sharing my screen here. Okay, so I'm going to start showing how you can you can find more information about our group, about the tools that we make. I'm going to show you how to locate the programs and how to download them, how to get them right in your computer. Then
Hey, Philip.
Yep.
Someone raised a hand.
I cannot see the chat window because I'm sharing right now.
Okay, allowed to talk. Dimitri, you can talk now.
It was a mistake. He put it in the chat.
Okay.
Okay, that's fine. All right, so, and then okay, so we're going to show how to run the programs, how to get them configured, and how to run them, and then we're going to show how to understand the output files. All right, so first we have a website here. It's called nativelab.org. So here you can find all the basic information about our group, especially the tools that we make. So we have a software tab here where you can find links to pretty much all the tools that we are developing right now. But most of them can also be found here on our GitHub account. So we have this GitHub page here. All the public projects are available here, so you can grab them, all of them on the GitHub, with the exception of Fragger, and I'm going to show you how to get Fragger. I'm going to show you here more specifically about FragPipe. So if you click, let me go back here. If you click here on the FragPipe repository, you're going to be redirected to our website where you can find pretty much all the information you need about how the program runs, some technical details. We have some tutorials, some workflows, some resources to show you how to read the output files, how to understand the workflows, and how to convert the files to the open format that we need. So pretty much everything can be found here. And if you click on the download button, you're going to be redirected to the place where you have to go to get the program. So now I'm assuming that everyone already has have the program in their computers. You don't necessarily need to follow the hands-on right now. So this is going to be recorded and broadcasted later. So you can just watch the presentation now. And if you want, you can try to follow up what you're doing later. Okay. So back to our GitHub, we also have links to other projects. So here, for example, this is the Philosopher website. You can find the source code. You can also find information on how to run the program, how to use all its options. And one important thing is, let me go back here to the FragPipe, is how to report back issues, or if you have questions or any requests. So here at the FragPipe website, we have this issues button here. And to use that, I believe you need to have an account. So you need to create a GitHub account. So if you click here, you can see every discussions that we're having right now, some open issues and things like that. And if you want to report a problem or ask for help or just get in touch regarding something in particular, you can click on this green button here called New Issue. And I'm not signing right now, so it won't open for me. But you have to click there, and then you will be able to report the issue you're having, or if you have any questions, or if you want to request for some kind of feature or something like that. So I'm not going to download the programs. I believe you already have them there. Some of them can be fetched on using FragPipe. And now I'm going to stop sharing here to allow Fengchao to share his screen. So Fengchao now is going to open the program, and he's going to load an experiment, and it's going to show you how to go step by step, configuring the program and executing and executing its options, so we can have the output files at the end.
Sounds good. Thank you, Philip. So, okay. So as Alex and Philip already presented, we have this FragPipe things, and I will show you how to use it with little effort to setting the parameters. So right now I have this latest released official FragPipe 17.1 in my computer. So you just go to the folder and go to this bin folder. If you are in the Windows, you can double click this one. If you are in the Linux, you need to run this shell script. So after
Fengchao, I think you need to share your proper screen.
Oh
I
yeah,
don't see
we are looking at your Google browser.
Oh, really?
Or the desktop, yeah.
Oh, sorry.
Okay, so yeah, just taking this as
Okay,
opportunity,
switch.
you can run FragPipe on Windows and Linux machines. We don't fully support Mac for technical reasons, but the program should be fully functional on any Windows or Linux version.
I'm sharing the right screen, right?
Yeah.
Okay, thanks. So, okay, let me say it again. So after you download the FragPipe, you will have this folder, and then you go to the bin folder here. So if you are on the Windows, you can double click this one to open the FragPipe, or if you are in the Linux, you can use this one. So after double click it, you will see the FragPipe opened. So initially there will be those three boxes will be blank, because license issues, you have to download the FragPipe by filling these informations and check the agreement due to the license issues. After you fill those boxes and click yes, FragPipe will download it for you automatically and put it here. And the philosophy is easier. You just need to click automatically. You don't need to fill anything. And Python, you need to have a Python installed, but it is not necessary. Python only needed if you want to build a special library, or if you want to use the split database function in the MS Frag. I will mention it later. So after everything is ready, you can go to the next tab, which is a workflow tab. And we have plenty of the workflows, preset ones for different kinds of applications here. The easiest workflow is the default one. The default workflow, they basically do the closed search using MS Frag, and then filtering the FDR using Philosopher, and that's it. There's no open search, no quantification, no DIA things. But for those for the other applications such open search, you can load the open search workflow, like click this one, then load. Then all the parameters will be set to the open search. It's almost ready to go unless you have some special requirement for your data. And then mass offset, if you want to perform mass offset search, as Alex mentioned, sometimes mass offset search is a better option. The open search, you can load this one too. After you load this one, you will see that in this mass offset box, there will be a list of the mass, which is from the common modifications. MS Frag will search all those common modifications.
Yeah, so just to open a parenthesis here. So this is probably the most important option in the program, the workflow selection, because FragPipe is basically a graphical interface that brings together all the projects that we do here, and it kind of orchestrates them in a way that the analysis can be done automatically. So instead of going tab by tab and program by program, setting all the options manually, all the parameters manually, we came up with this idea of offering predefined workflows. So knowing which workflow to load is probably one of the most important steps, because by selecting the workflow there on that tab, you pretty much configure all the tools in the program. Everything gets automatically configured. You can change the parameters if you need, and you can also come up with your own workflow if you want.
Okay, so there are really a lot of workflows, and I will not go through each of them right now due to time limit. Then I will use the one we are going to show for the TMT analysis, which is the TMT10 MS3 quantifications. After I load it, so here there is a global settings for the memory and the number of the threads FragPipe will use. So you can leave it as default. If you put it as 0, which means that FragPipe will detect free memory automatically and then use it. For the number of threads, it will default to the total number of the thread your computer have -1. So my computer have 12 cores, so it will use 11 threads. So then you can load the files, add the file. Right now we use one MZML file. It supports MZML or raw file. For the TMT, you should convert the raw file to MZML, but for the other workflow, you can use the raw file. Then I set the name of the directory as experimental name. Then we go to the database tab. Database tab basically allows you to specify the database, and then this is the one I have. It has.
Fengchao, sorry. We have a few questions here in chat. So some people seem to be having issues with EasyPQP. So let me see here. So apparently after the install, EasyPQP button doesn't work. So do they need to have Python installed?
Yes, I have needed to have Python installed. If you have Python installed, you can click this one. And in this version, FragPipe will freeze for a few seconds because it's installed EasyPQP. And then if you want to know if it's installed or not, you can click, let me do it. Okay, it will freeze. And then, yeah, it freezes. It's due to how God is dealing with this thing. And then after it's done, there's no message actually, but we added the message in the next release. You should be able to see it. But if you are not sure if EasyPQP is installed, you can open the Python command line and check. We have the GitHub tutorial I will mention later regarding how to set up the Python things, all those kind of things. I will mention later after I go through all the things.
Yeah, the Python library and EasyPQP are not necessarily for what we are going to do now. They might be necessary for DIA analysis and large analysis in case you don't have enough resources in your computer. But for now, they're not actually necessary. So you can skip those for now. And if you're having issues, we can talk in private later, or you can open an issue on our GitHub.
Yeah. So the next question is, even putting 0, the memory ratio still have the memory ratio. That may be because you don't have enough memory. But send us your error message so we can pinpoint what exactly the reason is. But most of the time, maybe your computer does not have enough memory. Yeah, it's a support FAMES data. You don't need to convert one FAMES raw into multiple MZMLs. We don't do that. You have to do that for MaxCon. We don't, but we don't need to do that for our tools. We can support FAMES natively, even with the raw file format. Don't even need to convert to MZML, but not for TMT, by the way. Yeah, it's free. For the academic usage, it's totally free. Even MS Frag is also free for the academic usage, but for the company, you need to buy the commercial license for MS Frag. But for the other tools, for the MS Frag and some other tools in the future, but for the FragPipe itself and the philosophy itself, it's free.
Fengchao, maybe we should read the question as well, because for the YouTube recording, I don't think
I'm
they have
sorry.
access to the chat. Yeah, so someone's asking if FragPipe is a free software or if it needs to buy a license. So yeah, as the Fengchao was mentioning, pretty much all the tools are open source and free to use, with the exception of Fragger, which will depend on where you are, if you're in a company or in your research institute.
So what is EasyPQP? Okay. EasyPQP is a Python tool from another lab. It is used to build a spectral library. And I will mention, this is basically about this tab. And I will show when to use EasyPQP when I mention the DRA workflow.
So Mark is asking, what is the ideal amount of memory? Well, yeah, that depends. I mean, we usually recommend starting with something around 16 gigs of RAM, but you know, some analysis. Well, we are going to show like this data set you are going to use. It's quite small, so you might not need that much. But if you have like a large fractionated wholesaleized, depending on what you're doing, you might need more. So it's kind of empirical, but we recommend starting with 16 to not have issues.
So can you send a? So another one asks, can you send again the link about the configuration? So sure. So okay, let me just go through these things. I guess it's more important. So okay, so when you go to GitHub, as Philip showed, when you search FragPipe, this is basically the first one in the search result. And then you can go to the landing page. Click this one, landing page. You will go to our FragPipe GitHub tutorial page. It have a lot of resources here. First of all, it will have the very basic resources about the using FragPipe. When you click this one, it will tell you very basic stuff, starting from configure FragPipe, which is the configure page I just showed. And there is also some other things about all those basic stuffs here, and you can take a look by yourself. And then there are also very specific tutorial about open search, massive set search, and some TMT related things, label free related things, and then all those how to interpret outcomes, outputs, all those list of the building workflows. And this is the setup. This setup is a little bit different from the setup mentioned here. Actually, I'm not sure why Sarah put it here. So this setup should be the one I showed before. And you can also go to this page by click this link, this hyperlink here in the FragPipe. You click this one to go to this setup page. So yeah, this GitHub landing page has a lot of resources, and you can learn basically pretty much over every almost all basic stuff by reading all those pages. And if you still have questions, and as Philip showed, you can ask a question here by creating issues. So yeah, we will normally respond very promptly. And then another one is, what does person 11 means in this case? Is it again related to memory? It is not related to memory. It is related to the number of the thread you are using. For example, in my computer, I have 12 cores in total. So FragPipe will use 11 out of 12. I leave one idle so that in case you are still needed to run something else. So when the MS Frag running, it will use 11 cores. When the Iron Quantum running, it will use 11 cores too. So this is what this 11 means. It is not related to memory, but more threads normally means more memory requirement. Okay, so dismiss. Okay, so yeah, let's go back to the TMT workflow. So after loading it, uploading it, I I go to database tab. So in database tab, it's where you specify the FAST file. We require the FAST file have both target and decoy sequences. If your FAST file only have target, you can click add decoy here. And then you can also add contaminations if you want. If after you click this one, it will automatically add decoys here. And then you can, after if you have a custom FAST file with target and decoy, and the decoy prefix is not the default one, you can click auto detect. It will detect what is possible prefix your FAST file have. In my case, it's still REV. So it will show you how many decoys are there. It's exactly 50%, which is what it should be. Then MS Flag tab, you really don't need to change anything unless you have some special requirement, because the workflow already set it for you. And then the validation, again, you also don't need to change anything unless you have a strong reason to do that. The PTM tab is for the open search and the glycol search, so it will not be used for TMT analysis. The MS1 will also not be used for TMT analysis. And then this is where the TMT quantification settings are. So the most critical part is this one. So you need to give or create an annotation file. Right now I have this annotation file ready. I can specify the path to it. If you don't, you can click this edit and create. So it will have its TMT templates. So it will have 10 rows, and then you can give name of your samples in, so you will be easier to track all your samples rather than look at those weird numbers and characters. Then rest of the settings, you don't really need to change, except if you have reference, you will need to specify the reference and then give the name of the reference channel.
Yeah, someone actually asked about this in the previous session. So here's the thing. We normally, when you have like 2 or more plexes, it's a good idea to dedicate one of the channels to serve as a reference, or some people call it a bridge. So it's usually a pool of all samples you have in all your experiments, and then that channel can be used by TMT integrator to during the normalization calculations. So that's why it's a good idea. If you don't have, some people sometimes don't have because of they cannot manage to lose one of the channels for a pool or a reference, for example. TMT integrator can also create what we call a virtual channel, which is basically an extra channel that will exist only for the purpose of the program to calculate the normalization across the different plexes. Yeah, so someone is asking, how does it works with multiple TMT plexes? Yeah, so what the program is going to do is it's going to load all PSM tables from all different TMT plexes, and it will merge them and apply the normalization and the filtering on all the data at the same time. And then the program can output the identifications in different levels. This is what Alexei mentioned before, as the gene level, the protein level, or the site level. So basically, the PSMs are rolled up to different. They can be roll up to the gene symbol or the protein ID, for example, and all the PSM quantification is summarized at that level. So, for example, if you do at the protein level, all PSMs that map to those particular proteins are going to be used. And if you do to the gene level, for example, all the PSMs that map to those particular genes are going to be used. And at the end, as we are going to show, we're going to see different tables. And those tables will have the index that can be proteins, genes, or peptide sites, for example, as rows. And on the columns, you're going to have the TMT labels from all the experiments. So someone is asking if we have an output. Yeah, so as Fengchao is going to finish the settings of the program, and then we're going to show how what the TMT tables, what they look like, and we're going to go through them in details. Okay, well, let me see the other questions. So Mark is asking about the cores. Yeah, those numbers are okay, like physical core 8, 16. Yeah, that's fine. That's usually what we use. Any standard good processing server is fine. In Alex's talk, he mentioned that the final step of TMT workflow converts back to absolute abundances. Can you explain what that means? For the calculations, the TMT integrator uses the reference channels, so it calculates a ratio of the abundances to the reference channel. So the final table will be a ratio value, and in one of these steps, which is optional, TMT integrator can convert back from a ratio value to an absolute value. Is this tutorial dataset available on the GitHub? No, it's not on the GitHub, but it's if you go to the May Institute website on the program listing, right next to this session, you're going to see a link called Docs. We added the links for all the programs and to the dataset on on there, so you can find there. Can I just use peptide lab? Yes. So Fengchao, can you click on the group by option there? Yeah, you see, so you can choose having a gene level, protein level, the peptide sequence, the PTMs, all. So if you click on all, you're just going to see all tables being printed. I use a FASTA file manually compiled with my target sequences instead of files downloaded from some databases. In this case, when I try to add decoys, nothing happens. So one of the things you should do first, if you have a custom FASTA database, and this is probably one of the most common issues we have today, the FASTA databases, they need to be properly formatted. So the headers, they need to be in a way that the programs can use them, because there are several elements in the headers that can be used by these different programs to find the protein ID or the gene ID or the description. So if you just write a custom format using your own format, it might be a problem. So what we suggest is go to the Philosopher website, the GitHub, and we have a tutorial or a description on how to properly format the FASTA headers. We normally ask people to follow the some open standards like the NCBI format or the UniProt format or the ensemble. You can have a very simple one that you can create, but you cannot add too many crazy elements on it because it might break the processing. So start by looking at what kind of format you have and check the website to see if the format is good. If it's not, you can always get in touch with us and we can help. Where do I get the appropriate FASTA sequence databases? So that, yeah, okay. So Fengchao, can you go back to the database? Yeah, so if you don't have a database, FragPipe can also download one for you on the fly. So let's show you how to do that. So this is Saccharomyces data set. So here we have some options. Yeah, you normally click, you can just click on the Saccharomyces over there. If the organism you're working is not there, you have to look for the UniProt ID of that organism, and that's something very easy to do. So you just go to the UniProt website, you type the name of the organism you're working, and right next to the entry that will appear on the website, there will be something called UniProt ID. So you can just paste the UniProt ID over there on that field, and then FragPipe will download the database for you, and we'll add decoys and contaminants. FragPipe only shows TMT 16. Does it support 18? Yes, I think this version that we are showing right now doesn't have TMT on the listing so far, but yeah, you can do the TMT 18 analysis. Well, in case, even though you the version that you are showing doesn't have the TMT 18, you can still load the 16 workflow, and then when you go to the TMT integrator tab, you can switch. You can change the number of channels on the annotation file, right? How do you add contamination? So when you download the database, if you download the database using FragPipe or Philosopher, one of the options that we just show on that small window that open is the contaminations. It allows you to add contamination. So we basically add 150 common contaminants, if I'm not mistaken. When you add FASTA, do you recommend add isoforms? In which case should I add it? Well, it depends, right? So it depends what you're trying to do. For a standard experiment, if you're not looking for isoforms, if you're not looking for variants, then we suggest not adding them. Isoforms and variants can complicate the analysis, especially the protein inference step. And they are usually good if you're doing explorative analysis and if you're trying to identify new variants, or if you're looking for specific variants that can be only present in certain samples. By default, if you don't know if you need it, you probably don't need it. So I would start without the isoforms unless you really have a reason for looking for them. The download button is not active on my FragPipe. What could be the reason? This is for the FASTA file download.
It's because you don't have philosopher specified. If you remove philosopher here, I guess it will be not activated. That's the reason. Please. So that's why you need to configure those three tools, at least those first two tools, to make sure that FragPipe have the tool. So make sure it can find the tool. So if I specify it, it will be able to download. Okay. So as to these questions, yes, it is the number of logical cores. Logical cores may be equal to physical cores, maybe not, depending on your CPU settings. If you have the like hyper-thread settings, it will be double, the number will be doubled. So this 11 is the number of logical cores minus 1.
You know, in theory, you can run any kind of analysis with one CPU core and like maybe 2 or 3 gigs of RAM. It's just going to take a lot of time.
Can you show how to add?
The more you have, the better.
In the official released version, we do not support TMT18, but in the future versions, there will be TMT18. I can show it right now. This is a pre-release version. We will release this month. So there will be TMT18 here. So, okay, I guess yes. No.
Actually, that's a good point, Fengchao. And this serves for everyone that is watching right now and for everyone that is watching on YouTube in the future. If you see or if you notice any difference between the version you have right now and what you are showing, that's because we upgraded the programs and because we are constantly adding new features and new elements, you might see differences. So there's nothing wrong. It's just because you probably have a different version from what you are showing right now.
So someone asked in the quant tab, I think it's alphabetical quant, right? Do we want to quant level to be 2 or 3? Looks like you have a set 2, 3, but the data looks like MS2 data, if I understand it correctly. It has MS3. Quantification is from MS3 and the identification is from MS2 in the data I'm going to show. So for this data, it basically have MS1 and then generate MS2 for the identification. Then it uses SPS MS3 to generate the MS3 level TMT intensities. So we use MS3. If your data has MS2 as for the TMT quantifications, you need to change it to 2. Or you can load the workflow here. For example, in the workflow, we have TMT10, which means that it's TMT10 and the quantification is at the MS2 level. We also have the TMT10 MS3, so which means that the quantification is the MS3 level. So it's really depend on your data.
Yeah, one other detail, since you came back to this screen here, there are a couple of buttons that are called Save Manifest. If you go back to the workflow, yeah, we usually recommend people to saving their experiments their settings. So if you click on the save manifest, that will save the list of files that you use on your experiment. That's always a good thing, because if you want to come back later and try to remember what you did. One of the files that it's going to be also available at the end is the workflow, which contains the settings for all the parameters used. Talk about normalization. Yeah, so Alex already mentioned a little bit about the process. It doesn't go much beyond that. So it's basically, we offer two methods. The most common one that we recommend for every process is the median centering, which is a very standard approach. But the most important thing I would say that is what the filtering that happens before the median centering, because one of the key parts that or things that TMT integrator does is to actually filter the PSMs before they get loaded. So things like overlabeling or PSMs with low purity, they all get removed by from the analysis. And also the selection of what kind of PSMs or peptides are going to be used for the quantification. So for example, we normally start with the unique plus razor, and that's what we recommend. If you want to really get the details about the mathematics of the normalization, we can provide. Yeah, we have something on the website, but we are currently working on a draft that will explain in details what's happening. So I guess, Fengchao, yeah, you can continue.
Okay, let me go back and load this workflow here.
Could you repeat how can we obtain the file in the tab of Quant?
It's not obtain. It's you can click edit and create here. So it will pop up a window, and then you can edit it here. This is how you, and then you click save, and then you can specify the location where you want to save, and then you will have this annotation.txt file.
The annotation file is basically a simple text file where you're going to have in each line the TMT channels or the iTrack channels separated by a space, and then the label that you want to add. If you don't have the annotation file, nothing's going to happen. It's just at the output, you're not going to have the label names. You're going to just have the channel names. So you're going to see like 136, 127N, 138N. If you have the file, the program replaces those channels by your labels. And if you don't have the file, you can click on that create button, and FragPipe will open that small window where you can fill the blanks, and they will be used on your analysis. Is there a way to add correction factors for TMT Quant? No, that's actually, we don't have that in place because we didn't notice any major changes for the quantification when they were being used.
Okay. So where are we now? Okay, fast file. So after I talk about fast file, I already go to this. So we have the annotation file ready. So then we go to the run tab here, and then you specify the locations, like, then you click run. Then you should be able to run the whole thing. And it will take a few minutes because the data is not very big for this data set. And then this is basically the TMT workflow and the TMT that we are going to show. Oh, by the way, if you want to use MS stats, you can click generate MS stats files here to let the philosopher to generate MS stats file. And then later on, you can use that file to give it to the MS stats and then run the R programs there. And then let me go through some additional workflows I guess some of you might be interested in. So let's go to the LFQ one. LFQ is label-free quantification plus a match between rounds. As Alex already showed in the presentations, we have the very fast label-free quantification tools named IronQuant, and we also have the force discovery rate controlled match between rounds. Everything comes with this workflow. So after you loading this workflow, you specify the files. Let me just randomly click some files as a demonstration. It's not important here. I will not really write. So when you select it, you might need to assign those files to different experiments. So there will be 2 columns for you to assign: the experiment name and the replicator name. The experiment name can be the empty, which everything will be in the same experiment, or alphabet and underline dash, alpha numerical and underline dash. For the bioreplicated, it can only be the integers. Yes. For this, someone asked, is the LFQ MBL workflow for DDA? It is for DDA. It is for DDA for the Orbitrap or Orbitrap with FAMES or DDA passive. It's supported actually all kinds of data I'm aware of. And you don't need to convert it to MZML. You can use a raw file or you can use .d from the DDA passive. So come back to the experiment. For the shortcut, you can click consecutive, so it will automatically assign each one to the experiment and 1, 2, 3, 4 bioreplicate, or by parent directory, which will all the file will be assigned to the experiment with the name of the directory, or by file name, whatever, or you set it manually. So I will click in this way. So since I've already loaded the workflow, then my first file is also ready. Then you don't need to change anything unless you have a strong reason to do that. So let us take a look at the MS1 Quant tab. This is basically the settings for the LFQ and match between rounds. Those are some basic settings, which the name explained by the name itself. And then this is the match between rounds, the RT tolerance, immobility tolerance, very straightforward. And the minimum correlations. Do we need to change the mass tolerance level? So mass tolerance is for the MF1 because it's label-free quantifications. It's not. There's always on MS1. There's no ambiguity on the levels. So match between round minimum correlations, as Alex mentioned, we correlate, given one round, we correlate to the calculate correlations of all the other rounds and then transfer the ions from the other round to the accepted rounds. So you can specify the minimum correlation so that only those rounds with the highly correlations will be transferred altogether. Or you can leave it at 0. And the top rounds is the HBR, MBR top rounds is also a similar function as the minimum correlation. You just control the number of the top rounds. The ions will be transferred, and then you can leave it as a very, very big number. Because we have the force discovery rate controls, so transfer the ions across all the rounds is not a problem. So here are 3 levels of the FDR. Someone asked, does match between rounds also transfer IDs between files designated with different experiment? The answer is yes. It transfers files among all those experiments, all those rounds, except unless you set something here. So it will give you a round to calculate correlations and only transfer the peptides among those with highly correlated rounds. Dimitri asked, but which PTM takes open search? Will be there, for example, modification characterizations for fast, something oxidation of proteins. I'm not quite sure if I understand this question. Maybe you can raise your hand and we can have a talk.
Are you asking about the workflows? Because you asked which PTM. Yeah, I'm not sure. Maybe you're asking about which workflow.
Yeah. Just raise your hand and or contact us after this conference, after this workshop. Another one sent us an error message. This error message shows that you are not using Java 64-bit version. So that's why when FragPipe try to use 5 GB memory, it cannot do it because you are using 32 bits version. 32 bits version only support up to 4 gigabytes. Please uninstall your Java and reinstall a 64 version. So someone sent another error message, said cannot find the purity column. Philip, can you answer this one?
Yeah, I don't know if you're using the versions that we are using from the currently release. We can so let's go through the all the way to the end with the processing, and then we can show the outputs, and then if we want, we can follow up with your problem.
Okay, someone asked where is the raw data located? I believe Philip have already answered it, right? It's
if you mean on the web, it depends. You mean on the website or the computer. If it's on the website, you should look for the May Institute program on their website. There is a docs link right next to this session with a PDF, and in the PDF you're going to see a link for the file.
let me see if I can get it. It's dot here. So here,
the
right?
last link, yeah.
Yeah, here. You can download it from Pride. In MS1 Quant, someone asked in MS1 Quant tab for top N ions, I see D43. Most people use a top 10 for DDA. In my experience, should we change to top 10? Yes, you can change if you believe top 10 is better than top 3. It's top 3 is experience numbers. There's no strong reason stick to top 3. There's a quick question. How do you tell if the run is done? You will know if it is done because it has all jobs done in 3 minutes, in 2 minutes. So yeah.
Well, if you have a slower computer, it might take more time, but you should see that message on the bottom over there.
Yeah. So yeah, as you can see, the job is done, and then the TMT thing is done, and I will leave it for Philip to talk about the result files later. Let me go through these workflows first. Okay, so go back to LFQ MBR. So we are here. We are left from here. So there are 3 levels of the match between run FDR controls. You can set it or use the default values. The top one options is for calculation of protein intensities. There are two ways to calculate protein intensities. So first way is top N approaches. Another way is the max LFQ approaches. For our experiment, max LFQ is always better than top N. But if you only have 1 or 2 experiments, then LFQ will have a lot of missing values because due to the algorithm of LFQ, max LFQ. So you might need to use top N. In the future version, if you check max LFQ, we will print both top N intensity and max LFQ intensity in the versions that we are going to release in a week. Then there are some. Then this is a tab. This is a panel about the label-based quantification. This is mostly for the phthalate delimitations or the other isotope labeling strategies. There it supports light, medium, and heavy. You can put in any labeling you want to use using this format from this tooltip shown here. In this example, it shows the phthalate, like the medium on K and R and the heavy on K and R. And then there are also advanced options, and you can check those meanings from the FragPipe tutorial web page I just showed. So after talking about this page, I think it's safe to go to the Run tab and click Run. I will not do that for now, but I will show you the result.
So let me just ask a few questions here. Okay. So yeah, okay. Dimitri and Gabriel are asking pretty much. I think they're related. Yeah, so something that we actually forgot to mention before is that on the Pride website, you're going to find the raw files, but you need to convert them to MZML first. So if you want to do the TMT quantification for now, using, that's one of the things that is going to be required for now on FragPipe, is that you need to convert the files to MZML first. And Fengchao was able to run it because he already has the files on his computers. And Lawrence, Lawrence is asking if we can label, if you can you have N15 labeling with 98, 95, 95, 99% heavy? I don't think you can, right?
We do not supporting nitrogen, heavy nitrogen or something. We only support isobaric and isotopic. And isotopic means silane or deamination, or those chemical labeling, not the metabolism labeling. Okay, so let's go to the TMT result. I have a TMT result somewhere. Not TMT, label-free quantification result. Since I just finished talking about the ARFQ workflow, so I have this small data set with 6 experiments. Each experiment has a one folder for it. It has individual result for this experiment. So this PNG file is basically the one Alex showed in the similar one as Alex showed in the slide. It's talking about the match between ROM FDR controls, the positive and the negatives. You can check if your data has a good separations, if you have some questions about your result. So the most important files basically should be those 4, and this mstats 1 if you want to use mstats. And those 4 is combined iron level, modified peptide level, and the peptide level. Peptide level means the peptide with the modification stripped. There's no modification, only sequence. And the protein levels. Take the protein level, for example. There will be some entries like the protein IDs, entry names, genes, all those basic informations, and then go to the number of the peptides and number of the spectral count. And the most important thing is intensity here. This data is run with the version that is going to be released. So you will see the intensity of the 6 experiment, also the max LFQ intensity. The intensity is from top N, and the max LFQ is from the max LFQ. This is from the future release. But if you are using the current release, you will only see intensity, which is from max LFQ or top N based on your settings. So this is basically what the combined protein looks like. For the peptide and modified peptide, it have the similar format. It's just have all the information such as the sequence, previous amino acid, next amino acid, start and end, length, charge, proteins, and some protein informations, and the spectral count of the peptides, and then comes to the intensity. Iron level is the same. And if you want to use mstats, there will be mstats file here. So if you want to use the reprint cryptome for the interact proteome, as Alex mentioned, you need to there will be 2 reprint files ready for you to use in the reprinter web server. You can use those 2 files, upload them to the server, and do the interactome proteomics analysis. So after talking about ARFQ, let me answer 2 questions here. Someone asked, what is the difference between top N and the max ARFQ? So it's different algorithm to calculate the protein intensities. For the top N, it basically take the top N of top N ions from this protein and then calculate the intensity, summation of the intensity, and use that one as the protein's intensity. For the max ARFQ, it use some more advanced algorithms, some kind of optimizations involved to calculate the max ARFQ intensity. You may need to check the literatures to know the details of the algorithms. There's 2 literatures that you can check. The first one is original max ARFQ paper from the Ulan Cox. You just search max ARFQ max quant, this paper, this MCP paper. we are talking about the original algorithms. And then we also detailed it, with more mathematical details in our ion quant paper. You can search these 2 keywords to go to ion quant paper. Here. Basically here is a calculation of protein titer use max LFQ. Here has all the mathematical details here, and you can take a look, and it will tell you how to do the calculations, basically. So someone asked, for PTM analysis, is there a place to specify variable and a statistic? Sure, it is. It's talking about specify modifications in the MS Frag tab. There will be modifications for the variable and the fixed. For the variable modifications, you need to specify site, mass, and the max occurrence, which controls the number of these PTMs in a single peptide. You also have the control of the maximum number of the modifications on the peptide. Also, the maximum combinations of all the modifications. For the site, it can be the amino acid sequence. If it's a protein terminal, you need to use these special characters as this yellow box showed here. Take a look at this yellow box, and you will know how to specify the protein terminal, protein C terminal, peptide N terminal, peptide C terminal modifications, and amino acid site modifications. For the fixed modification, it's already easier. You just change the mass value to a certain values here. It will be set. Okay, we have talked about the LFQ and match between rounds. Let's go to the open search. I guess some of you is interested in open search. So open search is also very easy. You just click open search and then load. That's it. Then you really don't need to change anything. And the special thing here is that the PTM shepherd, the PTM tab will be checked. This tab is specified for open search and glycoproteomics. With this tool checked, it will generate summarized tables of the modifications from your open search. I will show result here. I will show result later on. And then let's show it. Okay, I have open search performed just when Alex is giving presentations. I just perform open search. So those two, starting from global something, mod summary.tsv, and global something profile.tsv is the file I mentioned that generated by these tools. These two files summarize the detailed result of the modifications from open search. For example, this profile one have a detailed information of all those modifications, like the zero modification, which means there's no modification, there's no zero data. This peak means okay, let me give some basic information about open search. So the open search basically search the PSMs with a wide isolation window or with a wide precursor windows, rather than minus plus 20 ppm. It go from minus 150 to plus 500 Dalton. So you will see a lot of delta mass in your PSM tables, and those delta mass corresponding to the modifications from open search. It's some kind of unknown modification which you don't know before you perform the search. So with this delta mass, you can generate histograms. Then those histograms will have apex. We can do the histogram picking things. We will get apex. We will get a lower boundary of a peak and upper boundary of a peak. So the most abundant apex is 0, which makes sense because most of PSM don't have any modifications. Then it will come to the methylations, those unknown modifications, and then dimethylations, formulations, carboxylations, all those things found from open search. So this file will give a summary of all the modifications or data mass from your open search. Since I only put 2 ROMs in this data set, so there's not many found. If you have a big data set, you will see a lot of interesting things coming out. Okay, then it will have the number of PSMs, those mapped mass, those names of those possible modifications, and all those RT shift, all those kind of informations listed here. I will not go through them one by one. And then another mod summary file is like a summarized modifications from the previous file. It's just listed the modifications, names, and the mass, and the numbers from data set, number PSM from data set, and percentages. So the two file is kind of very useful for the open source because it will give you a basic idea of what kind of information your data have. With these files, you can cherry pick your modifications and do the more depth analysis with the following mass offset search or the closed search or whatever. So this is the thing I want to talk about talking about the open source. Okay, let me answer some questions. For the notation rule to dedicate isolation specifically, why do you need to identify for protein terminals, but when also add? Because those have two rules. This triangle is a marker. This bracket is specifically for the protein terminals. So this means protein. If I want to it at the, for example, like methylations or the methionines at the protein terminal, I can change this. So if I use this way, it means that so the modification only happens at the protein and terminal methionine, not all protein or terminals. So that's why this triangle needed to be there. It just tell you that it only happens at the protein terminal. For example, I can use other amino acid here. So yeah, so that's why I need to use two of them, not a single one. Someone asked, I got 2 errors on some test files I'm trying to run. For JNA could not load Brook library. Second, insufficient memory. So let's go through 2 of them one by one. So for the JNA things, please check the settings here. We have this paragraph here talking about if you are using Brook's native library, you might need to install these things. We are not allowed to distribute these things in our tools because it's a Microsoft library. So please download it and install it. And then insufficient memory, even though I have plenty of memory, well, just it's the I don't know the exact reason with this information. I needed to take a look at the log file or at least the original messages and the settings you have. And also remember to install 64 bits version of Java, not 32 bits version. Someone asked, is the search result output compatible with Perseus? Yes, it is. Perseus can take any tables, TSV, CSV. You just need to specify the major columns and the intensity columns you want to use. We actually have a tutorial of using Perseus for our result. Still, in this tutorial, in this GitHub, let me see here. Importing result to Perseus. When you click here, it will show you how to import FragPipe result into Perseus. After import the result, you can do whatever you want to do with Perseus. And someone asked, can you run MBR with TMT? No, we can't. But we will support it in the future version. But right now we can't. Someone asked, is there any documentation available explaining the columns of the open source result? Yes, it is. It is in the GitHub tutorial website and should be here. And yeah, so you see those 2 files are listed here, and I click it, it will tell you the columns, meaning of those columns. Yes. Someone asked, if processing multiple raw files in FragPipe in the result folder, will there be multiple TSV files or just one TSV for all raw files, and then result separated into different columns for different raw files in the TSV file? If you are talking about a psm.tsv, peptide.tsv, iron.tsv, and the protein.tsv, it really depends on your experimental settings. If you, for example, let's use raw file. If you don't specify anything here, everything will be in one single psm.tsv and other TSV files. If you put them into two different experiments, there will be two folders. Each folder have its own separate TSV. And in the root folder, there will be a combined TSV folder, TSV files, like I show here. I have 6 experiments. Each experiment folder have their own TSV file here. And then outside, there is a combined TSV folder here. So the number of the TSV files and how the data is organized in the TSV files is really dependent on the experiment structure you are specified here. Someone asked, is there any easy way to view MSM spectrum to confirm PTM containing open source? There is an easy way to view the MS2 spectrum in the future version. Okay, it's time to bring the pre-release again. We have this open visualization window function here. Let me show you, it should work. Yeah, it will load the data, load the TMT data I'm just using, and then it will show the annotations. But the tricky part is that for the open search, the modification is not, might not, depending on your settings, might not specified as variable modifications. So you will not be able to see them. You might not be able to see the modifications in your sequence. So the so it might be tricky to view the open search result, but for the closed search result, it should be easy.
So while you're loading there,
I think
Fengchao, let me just answer a few other questions here. So I see that Samantha and Silas are having issues with their program. So it might be hard for us to help you debug the issues you're having with EasyPicP right now. So what I suggest you to do is go to our GitHub, make an account if you don't have one, and please go ahead and open an issue describing what is the problem, and someone still today will be able to help you.
Could you talk something about the DIE workflow? Yes, I will talk about it after I finish this one. Let's go through it one by one. So regarding to the view result, there will be some there are some no bugs in the visualization tools. It's specified to a certain proteins. So I cannot view the other one, but I will try to view another one.
Fengchao, I don't think we are seeing the same thing you are seeing right now.
Wait, what?
We are not seeing the visualization tool. Are you watching something there?
Yeah, that's what I'm trying to do. Okay, here it is. So let me do it. Let me do it again. So after you run the FragPipe, everything is done. As I showed here, after everything, it's in the future version, by the way. The current version don't have it. After everything is done, you will have this path still specified. You just click this one in the future version. It will open another tool that you can view the spectrums. It's protein tables, PSM tables, experiment tables. There are 2 experiments in my demonstrations. So when you click certain proteins, it will show the PSMs found from these proteins. And then when you click one of them, they will show the annotated spectrums here. But it works very, very well for the closed search. But for the open search, since the PTM localization is kind of tricky for the open search, you might not see. It might have needed some tweakings to see the good result. Yeah. Okay, so let's I finished the open workflow. So okay, go to the DIA things. For the DIA, we have 2 sets of the tools that can perform the DIA analysis. So first one is DIA Umpire. It have published a few years ago that can generate okay, someone asked a question related to the visualization. Let me answer this in case. So yes, it is in the future version. The current one don't have it. Okay, the DI umpire. So the DI umpire is used to generate pseudo MS2 from the DI spectrums. With the pseudo MS2, you can search those files as a DDA data. And we recently developed the MS-Frag DIA. MS-Frag DIA can directly search the DIA data without pseudo generation or those kind of things. So let me show you with some toy examples. So first of all, let's load some fake data. It's not DI data, but it's fine. That's like fake data. Then let me specify the DIA spectral lab quant workflow and load it. Then in order to enable the DIA analysis, you need to specify your data as DIA. For example, suppose all data are DIA, you click set DIA, all data will become DIA. And if some of the data is from gas phase fractionated DIA, so you can select some of them and then set them as gas phase fractionated DIA. This GPF DIA data is mostly for library generation, not for quantification. So if you specify GPF DIA, those data will not be used in the later on quantifications. Only those DIA data will be used. Or if you have a data use that you don't need to include it in the library generation, but only for quantifications, you can set it as a DIA quant. So it will not be used for the library generations. It will be used in the quant later on. Also, you can specify DDI data. For example, there are DI, DDI, gas phase fractionated DI, and DI quant data. So the DDI data will be searched in a DDA approach, and they are used to generate a spectral library together with the DI data and the gas phase fractionated DI. For the other, someone asked a question about how about SWAT DIA science. Yeah, you can use it for the SWAT DI. Use the DI for the SWAT DI. It's just SWAT DI is DI data. Yeah. So after you specify the data types, let's make it easier. Let's do the DI first. Maybe half. Okay. Then your database tab is not important. So MS-Frag, there are not much you need to change for the MS-Frag. It will search DIA and DDA data separately, and then go to the FDR filtering, where in this page you also don't need to change anything. Then after the FDR filtering, you will have the PSMs, the peptides passing the 1% FDR. Then also then you send the why my EasyPQ? Okay, my EasyPQ is still also not available. I don't know why. Anyway, so you will see this page. Why my EasyPQ is not available? This is for the spectral library buildings. This is what the EasyPQP is. Okay, it is available now. So yeah, as I said, if you see this unavailable, you just click this one and then it will install the EasyPQP for you. Let me restart it. Because after install the Python library, you need to restart it. So let me use 17. Yeah, it's available now. So when this EasyPQP is available, you should be able to enable this tab. It will basically generate a spectral library using the peptide from the DDA or DIA data MS-Frag searched. Then there will be some settings. If your data have this IRT or CIRT, you can change the settings here. Then after this tab, it comes to the quantification tab using DIAM. We basically give DIAM the spectral library generated from EasyPQP, and then let Diane to do the quantification only. So here, Diane does not do the identifications. There's no spectral library prediction. There's no library generation by Diane. Diane simply do a quantifications using our spectral library. Then the good thing of using FragPipe over Diane is that you can generate a spectral library using DIA and the DDA, and also gas phase fractional DIA. So if you have different kinds of data like DIA, some DDA, and some gas phase fractional DIA, FragPipe will generate a big hybrid spectral library from all the data, and then use it as a template for the quantifications. So that's why that's the advantage of FragPipe over Diane. So after it's running, it will produce the result files. And the result file is pretty much similar to what I show here, except for some additional files from Diane quantifications. You will see it in your files. So this is about the using MS-Frag DIA to do the DIA data analysis. See another approach using the DIA umpire to do the DIA analysis. So this time, DIA umpire will be enabled. So DIA umpire will be used to generate the pseudo MS2 from the DI data, and then both DDA and DI data. The DI data means the pseudo MS2 from DI data will be used by the MS-Frag and do the database search in a DDA mode, because right now we already have the pseudo MS2 from DI data. We can perform DDA search for it. And then rest of the steps are similar to what I showed before. So the difference of this workflow and this DI umpire workflow and this DI spectral library workflow is using DI umpire or using the MS-Frag DI. From our test, MS-Frag DI give a better result than DI umpire. So let me ask some questions here. Someone asked, can the MS-Frag perform QC on spectral library? I'm not sure if I follow these questions. Can you elaborate more about performing QC on spectral library? What kind of spectral library are you talking about, and what kind of QC are you talking about? Yeah. Someone asked, what is a file type for spectral library? Can you append a pre-existing spectral library with new data, or do you have a rerun? No, you don't need. If you already have a spectral library, you can set them to quant, only for quant, no need for MS-Frag identification, no need for spectral library generations. And then go to Diane, specify your spectral library here. You can specify your spectral library here. And to the format, it's basically the format used by SpectralNut and Diane. And you can run small data and then let it generate the library.tsv. The name is library.tsv, and take a look at the format. Yeah. And someone asked, does the optional spectral library option select? Yes, it is. It select a spectral library here and used for that. Yes. Okay, so I think I pretty much finished DIA workflows here. And there are some others like the label workflows, site-like workflows. I will skip them, and feel free to have a look and try. If you have any questions, just remember, go to the GitHub page. We have all the basic stuffs here. We have the building workflows, list of building workflows, and it has some basic introductions here. You please feel free to take a look. If you still have some questions, please feel free to submit issues on our GitHub issue page. So, okay, Philip, can you talk about the result file, TMT result file? Now I think we only have half an hour left.
I think we have time. Yeah.
Okay. Yeah. Okay. I will let you.
Okay.
I'll stop sharing and let you to do.
Let me take over and okay. All right. So just to recapitulate a little bit, we are running a DDA TMT MS3 workflow. Okay, so where is it here? Okay, so we are using Saccharomyces database. Fengchao showed how to get Fragger configure here with the variable and fixed modifications. Pretty much everything else doesn't need to be changed unless you really know that you need those parameters changed. So if you don't know what they mean, you don't need to mess with them. We did the validation using, in this case, percolator here. So if you want to use peptide profit, you can just switch here the option. The program will run in the same way, but instead of running percolator, we run peptide profit. We run protein profit to perform the protein inference and to build the protein groups. And at the end, we performed an FDR filtering using Philosopher, generating the MSX file here. We perform MS1 and MS1 codification and the TMT codification using TMT integrator. And we got results. Okay, so I believe you can see my folder here right now, right? Okay, so there will be a lot of files generated in your folder when the analysis is done. So I'm just showing here the root folder of the project that Fengchao just executed. So we're going to see the database file and the binary representation of database file. We don't need to work with that. So here, this is the annotation file that we use, just in case you want to look again and see how the samples were annotated. So I'm just going to go through the most important files here. The top level files, we suggest you keep them because they can be used later when you're going through the publication, because they might contain important information about the experiments you're doing. Some of those files might be necessary when you're uploading your data to Pride, for example. So like, for example, which files were used. So here is just the list of files, that's the manifest. One of the most important things that you can have here is the log file. So we have this file here. It starts with log, and it basically contains the output from every single step and every single program executed by FragPipe. Here you can see all the parameters from all the tools. And this is especially important when you are submitting issues to us via GitHub, because that's pretty much the only way we can know what kind of analysis you did, what kind of which steps you ran or what workflow you executed, and we can debug in a better way the steps to try to find the problems you're having. But in overall, even if you're not having issues, this is one of the most important output files, so you definitely want to keep those, because they will contain the information about the analysis and everything else. Even later, when you're writing your paper, you might want to come back to this file here to understand what options you use, for example. Okay, so let's go to the results file. You're going to see two separate folders. Okay, the first one will contain the name of the experiment, depending on how you configure FragPipe, and the second one is called TMT report. Let's go into the first one. So here we have again the annotation file. We have a text representation of the FDR filtering. So here we can see how many PSMs were considered for the FDR filtering. We can see how they were broken down into these PSM, peptide, and ion layers, showing their individual FDR scoring, the number of proteins that were identified by the protein inference, and the current number of target proteins that were approved under here in this case 1% FDR scoring. The second step for the filtering, and the final numbers here, showing that for this experiment, we got about 10,000 ions, 9,000 peptides, 1,400 proteins, and 11,000 PSMs. So all this is important later if you want to understand how well the analysis performed, or if you want to keep these recorded somehow. So we just suggest you keep everything. These files that end with pepxml or pin or just in XML, these are files that were used by the programs to perform the analysis. Depending on what you're going to do later, you might want to keep them, especially if you want to submit data to Pride, for example. But you don't need to inspect them. They will not be informative unless you're doing something more detailed. Now, the most important ones are these one here that ends with a TSV on it. So basically, all the data from the experiments are broken down or separated in what we call layers, and they are filtered individually. From the PSMs, we assemble protein lists, peptide lists, and ion lists. Okay, so these tables, they look very similar to each other. Some might be more important than others. You might use just one, or you might use all. It depends on what kind of experiment you have and what you're trying to do. I'm going to show you the two most important ones that people usually use the most, especially the PSM. So let's open the PSM table. Okay, so this is basically all PSMs that pass the 1% FER filter. I'm just going to go really quickly through the file itself, showing what kind of information we report here. You don't need to take notes or anything like that. If you miss anything, don't worry. We have on the website that we showed before, that Fengchao also showed again, all the columns of every file we have on these pages explaining what they mean, what kind of information they have. Okay, so basically here we have PSMs in different rows, and we try to put as much information as possible about their identifications. So we have information about the PSMs itself, the sequences, the charge, the retention time, all the calculated and observed masses of the fragments and the peptide sequences. We output as well all the scores that were calculated by the search engine and by the validation tools, either peptide profit or protein profit. You can also see, so here we have the MS1 intensity, the assigned and observed modification. So the difference between assigned and observed is basically if the modification was defined on the parameter file for the search engine or not. If you define the modifications as a variable or fixed, we call them assigned, because you basically assign those on the search for the search engine. And if you are doing open searches, you're going to see this column here filled with all kinds of delta masses. So we call them observed because the search engine was able to find them by just looking at their sequence. We have a question. For TMT workflow, do you have parameters to take into account co-isolation, interference, and SPS threshold? Well, one of the things we use here, and I think it might be related to what you're asking, is this purity score. So PD gives you something called interference, an interference column, which goes from 0 to 1, like a percentage. This purity score that we have here is basically the same thing. So we look at the precursor spectra to see how much interference we have from other peaks. So you see here it goes from 0 to 1, 1 being like 100% pure, meaning that there is no co-isolation or anything else. This is one of the things that TMT integrator uses. I believe that the standard threshold is 0.5, so everything with a purity of 0.5 or higher is used. The other ones are discarded. Let me see another question. Is the default workflow compatible with DDA, PRM, SIM data? I think you mean the default workflow in the FragPipe. I'm not exactly sure. Maybe Fengchao can answer you, but
yeah.
I believe
The
DDA, right? But I'm not
DDA
sure
is
about the
for
other
sure.
ones.
PIM and SIM depend on what kind of analysis do you want to perform. If you just want to perform the peptide identifications, yes. For the quantifications, it really depends on what do you want to do.
Yeah.
I mean, you because for the PIM or SIM, you can do the quantification for the MS2 level or MS1 level or together something. Yes, go to the. It really depends. Yeah, I'm talking about the default value is closed search. It's just do the identifications and yes, default workflow even don't have the quantifications. So default workflow should work for the DDA, PIM, SIM things. But if go to the quantifications, if you want to use MS2 quantifications for the PIM or SIM, our tool cannot do that. But if you want to use MS1 quantification, yes. It's not about a TMT.
Right,
it's not about TMT or it's not about a PTM.
All right, so so yeah, we have this purity score. We also have this unique score that tells if the PSM is unique, and unique here means if the peptide sequence is mapped into different proteins, more than one protein actually. We have this session here that tells more about the identification itself. So the proteins that were mapped by those PSMs, the RDs, the entry names, the G name, and this comes back to what I mentioned before about having a good FASTA database. If you're using a FASTA database formatted with one of the common headers that we see as in UniProt or as Ensemble or NCBI, all this will be filled with information like this. Otherwise, it will be blank because the program cannot figure out what kind of information you have on the headers. And here at the very end of the file, we have the information about the TMT channels. So here are all the labels, all 10 channels and their intensities. So this is the PSM table. Okay, it's I guess pretty much the most important one that is outputted by the pipeline. We have an alternative version of the PSM, which is the MS-stats CSV file, which is basically a version of the PSM table formatted in a way that is already it's ready to be consumed by MS-stats. I'm not sure later if Olga or team were going to use these outputs on their tutorial, but I think they're probably going to use it. So it's basically the same PSM table, but just with a different format. So you can import directly into MS-stats. And the other table here is the protein table. So this is basically the information of all the identified proteins. So here, for example, you can see like some of the contaminants that are with identifier. These are all target proteins. So the basic information about them. If you're using data from UniProt, for example, you can see their evidence level from HUPO, their description, and here some quant data. We tell how many peptides map to those proteins. We divide them by total, unique, and razor, meaning that total is just like the whole summation of all peptides. Unique is just the peptides that map uniquely to that protein. And razor is unique plus razor. So meaning that you're going to see unique peptides plus the shareable ones that got assigned exclusively to this protein. Same thing for spectral counts, if you want to check the spectral counting. And the MS1 intensity as well. The list of all modifications from all peptides mapping to that protein. And here at this and very end again, the TMT labels with the intensities. Okay, so this is what we call the raw output, because there is no normalization, there is no filtering. It's just what comes out from the FDR filtering, right? So all these numbers, all these quantifications are just raw because there is no processing done on them. Now, if we come back here to the output folder, we're going to see that there is one extra folder here called TMT report. And this is where we can find the TMT integrated output tables. So and here again, we can see genes, proteins, and peptides being quantified. And you might ask, okay, which one should I use for my downstream analysis? We use the TMT integrated outputs when you are dealing with the TMT or iTrack data because they are already normalized. But if you are trying to do something different, for example, if you don't want to use TMT integrator, or if you want to use MS stats TMT, for example, you start with the MS stats table, which is not normalized. Okay, another question. Sorry, I could be wrong, but I thought both DIA and PRM use MS2 for quantification. So FragPipe can do DIA quantification by DIA NN and Empire, but not for PRM data.
Let me answer this one. So correct and wrong. 答案 only support DIA data and it do the MS1 and MS2 altogether for the DIA data. But since as you can see, FragPipe using DIA to do the quantification for the DIA. And we use IronQuant to do the MS1 level quantification for DDA. So there is no place for the quantification for PRM data. That is why we cannot do the MS2 level quantification for PRM data. But if you only want to do the MS1 level quantification for PRM data, you can treat it as DDA result and give it to IronQuant.
All right, thank you. Okay, so let's take a look here real quick about over this table. So here right away you can see two set of tables. We have like abundances and ratios. So it basically it's the same data, but the quantification on the first set is the numbers are actually ratios to the references, and on the second set they are just the numbers were converted back to the absolute abundances. And we have here them MD because we use the median centering normalization. If you use another method, you're going to see a different name there. And we have gene, peptide, and protein, and gene, peptide, and protein. That's because we don't have phospho data or any kind of PTM analysis data, so we don't see the phospho sites or PTM sites, for example. So the structure of these tables are pretty much the same, and these are like expression matrices that you see, very similar to expression matrices that you see from RNA-seq analysis, for example. So let me show you what I mean by that. So this is the gene level table. We have as an index gene symbols. Okay, they are unique here. We also include here the number of PSMs and some other information about them, like the number of PSMs, the ID of the protein, the probability, and the reference channel here. And here on all the other columns, you see the normalized TMT abundances. Now, if you have, let's say that you have two TMT 10 plexes in this analysis, you'd have on this table here 20 columns with the abundances. If you are analyzing 10 TMT plexes, you're going to have like 100 columns. So all the experiments, all quantification from all the observations from all experiments are going to be summarized in this single table, right? And their labels are going to be as you pointed on the annotation file. So this is a pretty simple table. It's just this. It's just the indexes and the quantification. They're negative because of the way how we normalize. I don't recall exactly how the calculations they work right now to make them look like that. But when you observe them as if you do like a box plotting of, well, I think they're negative because I opened the ratios, right? Yeah, there you go. They're negative because of the ratios, but that's because of the adjustment to the reference channel. These channel values are normalized with the reference channel. Yeah, so these are normalized using an internal reference. And so yeah, we were looking at the ratio. That's why some of those are negative. This is the absolute values. So it's pretty much the same table here. We have the genes symbol as indexes and the quantifications in columns. So let's look at the protein here real quick. And so the protein, you have like the protein ID, it's the section number. Depending on what kind of FASTA file you have, this might be different. We also include the G name here. I think in this column, they might not be unique depending on the settings on Team Integrator. And here also the abundance of values. And that's it. That's basically the tables that we use. Those are the outputs we use. And as Fengchao mentioned, if you have more than one experiment, either Team T or iTrack or label free, you're going to see an extra set of tables on the upper folder, upper level, which is basically the combined list of all peptides and proteins that you observed in these experiments. So I guess that's it. That's what we had to show you. If you still have questions, please take a look at our website again. Check if you can find the answer to your question there. If you have more questions about these tables and their formats, you can also look at the website. We have like description of them. If there is anything out of place or you're still confused, you can always open an issue. It doesn't need to be like a bug report or anything like that. Some people just open issues to ask questions. That's fine. And then we can get in touch and help you. Do you have anything else to show or to talk about, Fengchao? No? Okay. So if no one else has any other questions right now, I think we can wrap up. Yeah, okay. All right. So I don't see any other questions. So I'm going to stop the recording, stop sharing. And I don't think Olga is coming back right now, so we can just adjourn for now. And yeah, thank you for your time, and thank you for staying